Hi viewers, in this class we are going to discuss the ELISA apart from the antigen antibody reactions. ELISA, under this topic we are going to discuss introduction to ELISA, types of ELISA tests, direct ELISA, indirect ELISA, sandwich ELISA, competitive ELISA, and the advantages of ELISA test, applications of the ELISA test. ELISA, abbreviated as Enzyme Linked Immuno Sorbent Assay. Enzyme Linked Immuno Sorbent Assay. This is a solid phase immune assay. This assay is used to measure the amount of either antigen or antibody. The presence of antibodies bound to antigen absorbed onto the plastic may be detected by the use of anti-immunoglobulins labeled with an enzyme. This can be measured by the appearance of a color on addition of proper substrate, so that it is called enzyme linked immune sorbent assay. Here you will observe the enzyme linked immunosorbent assay is also known as EIA, enzyme immune assays, is a test that detects and measures antibodies in your blood. This test can be used to determine if you have antibodies related to certain infectious conditions. Antibodies are proteins that your body produces in response to harmful substances called antigens. ELISA is a popular format of wet lab type analytic biochemistry assay that uses a solid phase enzyme immune assay to detect the presence of a substance, usually an antigen, in liquid sample or wet sample. In 1971, there were the two groups of scientists in 1971, Peter Pellman and Eva Engvall at Stockholm University in Sweden, and Anton Schors and Bach van Wiemen in the Netherlands. They independently developed the enzyme immune assay, otherwise the enzyme linked immunosorbent assay. Here, antigen of interest is absorbed onto plastic surface. It is a sorbent substrate. Antigen is recognized by a specific antibody, that is Immuno. This antibody is recognized by second antibody, which has enzyme attached, enzyme linked, and substrate reacts with enzyme to produce product usually colored. So that it is known as the enzyme linked immunosorbent assay, that is the ELISA. And see the principle behind the ELISA test. We will use an enzyme to detect the binding of antigen and antibody. The enzyme converts a colorless substrate, which is usually a chromogen, to a colored product, indicating the presence of AG or AB binding. An ELISA can be used to detect either the presence of antigens or antibodies in a sample, depending how the test is designed. ELISA was developed in 1970 and became rapidly accepted by all the scientists. And there are different kinds of ELISA. The enzymes that are used in ELISA include beta-galactosidase, glucose oxidase, peroxidase, and the alkaline phosphatase. The ELISA technique mainly based on antibodies, and some antigens can absorb to wells of the molded polystyrene plates without losing their immunologic capabilities. Antigen and antibodies can be linked to enzymes to form complexes that are immunologically or enzymatically functional. So there are different kinds of types of ELISA. namely direct ELISA, indirect ELISA, sandwich or double antibody ELISA, competitive ELISA. These all are the different kinds of ELISA tests. Let us see one by one. First, see the direct ELISA. What we will do in the direct ELISA? There are different steps in the direct ELISA. The first one is a buffered solution of the antigen to be tested for is added to each well of a microtiter plate, where it is given time to adhere to the plastic trough charge through the charge interactions. And second one, a solution of non-reacting protein such as bovine serum albumin or casein is added to well. So usually each plate contains 19 wells in order to cover any plastic surface in the well which remains uncoated by the antigen. The third one is the primary antibody with an attached conjugated enzyme is added, which binds specifically to the test antigen coating the well. And fourth one, a substrate for this enzyme is then added. Often the substrate changes color upon the reaction with the enzyme. That will be read in calorimetry. Otherwise, then the higher the concentration of the primary antibody present in the serum, the stronger the color change. Often a spectrometer is used to give quantitative values of the color strength. So like that we will detect the direct ELISA. Then coming to the indirect ELISA. In the indirect ELISA, what we will do, antigen is absorbed to a well in an ELISA plate. Detection is a two-step process, where in the first one, an unlabeled primary antibody binds to the specific antigens. Second one is an enzyme conjugated with the secondary antibody that is directed against the host species of the primary antibody is applied, and thereby again we will add the suitable substrate. And if there is a antigen antibody complex, that if it is detected by the enzyme conjugated secondary antibody, then substrate, upon the action of the enzyme, it modifies its color, then again it will be calculated, otherwise measured. So it is indirect ELISA. And then coming to the third one, so indirect ELISA steps are: antigen is absorbed onto the microtiter plate, add serum to test for antibody, add secondary antibody, primary antibody binds to secondary antibody, add substrate which reacts with enzymes and change color and show the antigen strength that will be read by the spectrometer. Then coming to the sandwich ELISA or double antibody ELISA. So in between the two antibodies, the antigen is present like this, like a sandwich. Here, the sandwich ELISA is used to detect a sample antigen. The steps are as follows, where a surface is prepared to which a known quantity of capture antibody is bound. Second one, any non-specific binding sites on the surface are blocked. Third one, the antigen containing sample is applied to the plate and captured by antibody. Fourth one, the plate is washed to remove the unbound antigen. Fifth one, a specific antibody is added and binds to antigen. This primary antibody could also be in the serum of a donor to be tested for reactivity towards the antigen. Then sixth one, enzyme linked secondary antibodies are applied as detection antibodies that also bind specifically to the antibodies FC region. Antibodies FC region, that is a non-specific one. Then the plate is washed to remove the unbound antibody enzyme conjugates. Then a chemical or substrate is added to be converted by the enzyme into a color or a fluorescent or electrochemical signal, whatever it may be. Then that will be the absorbance or fluorescence or electrochemical signal of the plate well is measured to determine the presence and quantity of the antigen. So that will be measured. So these are the steps involved in the sandwich ELISA. So here it is looking like the sandwich. At the bottom there is antibody, and above there is antibody. In the middle we will see the antigen. And coming to the competitive ELISA. The competitive ELISA, what we will do, first one, what we have to do is, an unlabeled antibody is incubated in the presence of its antigen, that is in the sample. Thereby, these bound antigen antibody complex are then added to an antigen coated well. The plate is washed, so unbound antibodies are removed. The more antigen in the sample, the more antigen antibody complexes are formed, and so there are less unbound antibodies available to bind to the antigen in the well. Hence, it is a competition. Then thereby, the secondary antibody specific to the primary antibody is added. The second antibody is coupled to the enzyme. Then a substrate is added, and remaining enzymes elicit a chromogenic or fluorescent signal. The reaction is stopped to prevent the eventual saturation of the signal, and thereby that will be measured. So simply, here the competition occurs between two antibodies for the same antigen. It has been used for detection of HIV antibodies. The microtiter plate wells are coated with HIV antigen. Sera to be tested is added to these wells and incubated at 37 degrees Celsius and washed. Then antigen antibody reaction occurs. That will be detected by the enzyme bound secondary antibody. Then what are the advantages of ELISA? Reagents are relatively cheap and have a long shelf life. ELISA is highly specific and sensitive. No radiation hazards occur during the labeling or disposal of waste. Easy to perform and quick procedures. Equipment can be inexpensive and widely available. ELISA can be used to detect the variety of infections. These are the advantages of the ELISA when compared to remaining antigen antibody reactions. And finally, the applications of ELISA. So we will use ELISA to detect the hormones, proteins, infectious agents like virus, bacteria, parasite and fungal infections. And you can also detect the drug markers, tumor markers, serum proteins, and you can also see the vaccine quality control. And also will use for the detection of the genetically modified organisms, GMO, and for all the rapid tests to detect the IgG, immunoglobulin M and immunoglobulin A, and also for the newborn screening, and also it is widely used in the clinical research. So these are the broad spectrum of applications of ELISA. So by this, we have learned what is ELISA and how many types of ELISAs are there and how they will be conducted in the laboratory. So, and applications as well as the advantage of the ELISA test. Thank you all.